Now, the other main class of testing, we said there was genetic testing. The other main class is immunological, or otherwise known as serological testing. Serological testing takes into account antibodies and antigens. And I hope you guys are familiar with these terms, but just in case, antigens are the bad guys. The antigen is a foreign molecule, like a protein from a bacteria or a virus. So the foreign invader is the antigen. And the antibodies are proteins made by the immune system in response to the antigen. So for example, if you have a viral particle that has spikes on the outside, as the immune system responds to this virus, it's going to make these Y-shaped proteins that will bind to the virus. And the part that binds is the arms. The arms of the Y is going to bind to the antigen. And when the antibody binds to the antigen, this will target it for destruction by the immune system, which we'll talk about later this semester. So the only way somebody would have antibodies against a specific antigen is if they were infected with that antigen. Somebody would never have antibodies against the flu unless they were infected with the flu. Somebody would never have antibodies against HIV unless they had been infected with HIV. All right, so antibodies are made in response to an antigen. And of course, if somebody had been vaccinated against the flu, then they might have antibodies against that strain of flu. So there needs to be some sort of antigen present in order for the immune system to make antibodies. So antibodies, measuring them, is a way for us to say, has this person been infected with this thing? Yes or no. How we measure antibodies and antigens, there's many different methods, but the one you're going to see, I guarantee you're going to see this, that's why I'm telling you, wouldn't be wasting your time, is called ELISA. And the ELISA stands for Enzyme-Linked Immunosorbent Assay. Very, very common technique used to diagnose infections. We use ELISAs for pregnancy tests, which is not an infection, but I just wanted to throw it out there. Pregnancy tests use ELISA. We use it for HIV, Zika, chickenpox, hepatitis, Lyme disease, and the other tick-borne illnesses, syphilis, toxoplasmosis, and almost everything else. So we use this test to diagnose just about everything. So let me tell you how the ELISA works. There's two main types of ELISA, but both of them use these plates that you've seen before, the 96 well plates. They're also called microtiter plates. And I know you've seen these before because we used them in BMS 103 lab. So the first type of ELISA is an indirect ELISA. Let me explain how this works. So in this microtiter plate, there's a bunch of little wells, and each well we would do a separate ELISA reaction. So we're going to zoom in on two wells here, okay? So here's the first well, here's a second well. And the example I'm going to give you is, let's say we're testing somebody for the HIV virus. In this side here, side 1, patient 1, we don't know it yet, but I'm going to tell you this person is positive for HIV. And we'll say over here in well 2, we don't know it yet, but I'm going to tell you this is an example of somebody negative for HIV. The first step is we're going to add into the well, we're going to add the known HIV antigen. And when I say known, I mean I grabbed it out of the freezer. Okay, we already had this antigen prepared in the freezer. It's a standard antigen used in this test every day. So we just grabbed it and we're going to put it into our well. And what's going to happen is the HIV antigen is going to stick to the bottom of the well. Okay, so the red here, this is the HIV antigen that I threw in there. Our next step is now we're going to add the patient's sample. So if we were testing HIV, we would do a blood sample. So we would we would do a blood test. We would take blood from the patient and we would filter out things like red blood cells and platelets. We don't need that. We need just the serum that contains the antibodies. Now, there's a plethora of antibodies that's in the serum. It's not like there's just one, there's many. But remember, antibodies are really specific to their antigen. So patient 1 here, we already know is positive. This patient is going to have antibodies against HIV. What's going to happen is those antibodies are going to stick to the HIV antigen. Because remember, only if somebody has been exposed to that antigen will the immune system respond by making antibodies. Patient 2 is not infected with HIV. They don't have any antibodies against HIV, so nothing's going to happen here. There's going to be nothing that binds to the antigen. Now our third step is we're going to add a known secondary antibody. When I say known, I mean I'm going to pull it out of the freezer because it's a standard thing. I bought it from a company. The secondary antibody will only bind to HIV antibodies. In this first patient, I'm going to add the secondary antibody. It's going to bind to the primary patient's antibodies. It's going to bind to it. But in patient 2, when I add the secondary antibody, there's nothing for it to bind to. So it's just going to be kind of floating around. And eventually it's just going to wash out, right? It's going to be gone. It's going to wash out of the well. Now, linked to the secondary antibody, there's an enzyme. Because remember, ELISA stands for enzyme link. And there's a special little enzyme that's going to be attached to the secondary antibody. This enzyme will change color when I add a special substrate. So my last step is I'm going to add a color-changing substrate. The substrate's going to interact with the enzyme. It's going to turn the whole sample a different color. Whereas in my negative sample, there was no substrate present, so it doesn't change color. So the main takeaway, what you're going to see, if the individual is positive for whatever you're testing, you're going to see a color change in the well. It's not always yellow, but it could be yellow. And if the patient's negative, you're going to see that the liquid remains colorless. There's no color change. So this is a really easy way, based on whether the patient's serum contains antibodies against the antigen or not. This is a good way to test if the patient's positive or negative for those antibodies. Here's a pretty picture showing the steps again, in case you like prettier pictures than my bad drawings. So here's an example of what you might see. If you see the well has changed to yellow, that would indicate a positive result. If the well remained colorless, that would indicate a negative result. Now notice that some of the wells have a lighter yellow and some of them have a really dark yellow. What this would mean is a difference in what we call the antibody titer, which can tell us a lot. A lot of times, if there's a high amount of antibodies, a high titer, you would see like a darker yellow. This could indicate a couple things. Could indicate the patient's been infected longer, so their bodies had more time to make a lot more antibodies. If it's a case of a latent virus, if there's a high antibody titer, it could be an outbreak period. Whereas if it's lighter, maybe the patient's in dormant stage, so they don't need a lot of antibodies. So a low titer may indicate a dormant period or a recent infection. Let me show you an example. Remember I told you last class, pictured in blue here, HIV goes through many stages of infection. This is days following the acquisition of HIV. With HIV infection, about 10 to 14 days in, we see a really high spike in viral load, pictured in blue here. The viral load shoots up really fast. The body takes a few days, maybe a couple weeks, to be able to respond to this high viral load. So what you'll see with HIV is the antibody titer takes a little while before it can catch up. Once the antibody titer catches up, gets higher, then the viral load will decrease as antibodies destroy the virus. So again, if we look at the titer, the antibody titer, that can tell us some clues as to what stage of infection a person's in. Now, similar to the last ELISA, we could do the ELISA the other way around. This would be called a capture or sandwich. I usually call it sandwich ELISA. How this would work is instead of measuring antibodies, we measure for antigen in the patient. So in the indirect ELISA that we talked about before, we're measuring patient antibodies. Whereas in the sandwich ELISA, we're not measuring patient antibodies. Instead, we're measuring if there's antigen in a patient sample. So how this would work is we've put a known antibody, meaning an antibody that I ordered from a company and I pulled it out of the fridge. So I put the known antibody in first. The antibody will stick to the well. Then I add the patient sample. If the patient has active antigen in their sample, like active bacteria or viruses present, the antigen will stick to the antibody. And then I add unknown secondary antibody that I pulled out of the freezer. I ordered it from a company. And the secondary antibody will only bind if the antigen's present. So it'll create like a sandwich where you have antibody, antigen, antibody. And then I put an enzyme on my secondary antibody that'll change color. So again, positive, you would see a color change in the well. Where this might be useful is if the patient's really early in infection, like remember in the last chart, if the patient's really early in the stage of infection, they don't have antibodies made yet because the immune system takes time before it starts pumping out antibodies. But the viral load is still high. So where we would use a sandwich ELISA is if there's a high viral load, and we suspect there's a high viral load, but we suspect there might not be antibodies yet. So we could use this method in that case. Now, the very last thing I want to tell you about today is something that's become a trend in the healthcare field. And I'm sure you guys might be able to tell me more about this. Why is this such a trend? I want to know. The trend is what's called a point of care test. And you guys might have heard it also called like rapid test, like a rapid strep test or a rapid coronavirus test. There's many examples, like pregnancy tests are rapid point of care tests. Flu, they have ones for flu now. They have HIV. They have strep, which has been around for a while. And they have COVID-19 rapid tests. Here's how rapid tests work. They're very similar to the ELISA, only what you do is you take the strip. Picture like a pregnancy test. This is where you're going to add the sample. So this could be a fluid sample like urine, or it could be a throat swab, or it could be like a saliva, could be blood, a drop of blood, right? So what happens is if the patient has antibodies against whatever you're testing. So let's say we're testing for HIV again, just because we've been using this example. So if the patient has antibodies against HIV, the antibodies will come into the nitrocellulose membrane and they flow in one direction. It always flows it's created to flow that way. So what will happen is there's these gold particles. It's not always gold. Sometimes we use like a colorimetric method, like a blue. There's these little gold molecules and the antibodies will bind to the gold. Some of the gold molecules are free and those are the ones that the antibodies are going to bind to. And some of the gold molecules are attached to a control antibody pictured in purple here. This is control. All right, so the gold molecules move along and they hit the test line. And on the test line, you have the antigen pictured in this little green ball, the known antigen that's ready to go on the test line. If the patient's negative, they don't have any HIV antibodies, nothing will bind. There's nothing there to bind. So the gold will just keep going. But if the patient's positive, the antibody will stick to the antigen and the gold molecule will get stuck there. It gets stuck. And then some of the antibody will keep going. And in this case, all of the gold molecules will keep going. And then you hit a control line. On the control line, the control antibody that was already there is going to bind with the gold in both the negative and the positive sample. And then everything just keeps flowing till the end. So what you would see if the patient's positive is you would see a gold or a color change on the test line, and you would see a color change on the control line to ensure that the test worked. If the patient's negative, you see nothing, no change on the test line, and of course the control line, you should see a change. Yeah, this is all fun and dandy, right? But the problem is these tests are very prone to giving both false positives and false negatives. I'm not sure what the trend is here. Is it because they're easy to do? Is it because pharmaceutical companies are pushing these out to make money? Is it because people are living in a rapid society where you need to know everything right away? And these tests are never a substitute for real immunological or genetic testing. But I want to hear what you guys have to say about them because you're the ones that will be maybe using these, right? Okay, so that's all I'm going to say today about diagnostic testing.